Welcome to the second half of the urinary system, where we will finish up the renal functions, specifically reabsorption and secretion, and then look at the elimination of urine from the body and do a bit of urine analysis. So far, we've looked at one of the major processes of the nephron, which is glomerular filtration. So that creates the filtrate or tubular fluid that is collected in the capsular space of the glomerular capsule or Bowman's capsule. That fluid will then move into the tubular system and undergo reabsorption and secretion to modify the contents and form urine, and in doing so alters the composition of the blood. Tubular reabsorption is the process of returning most of the filtered water and solutes back to the blood so that they are not lost in the urine. This includes glucose, amino acids. Urea, we know, is a waste. It is passively reabsorbed and will have to be actively secreted. Ions include sodium, potassium, calcium, chloride, bicarbonate, and phosphate. Vitamins are reabsorbed, and even small proteins and peptides that escaped the filtration membrane can be absorbed. So by the time the kidneys are done, 99% of the filtrate is reabsorbed, and most of this reabsorption occurs in the proximal convoluted tubule. Tubular secretion is the transfer of material from the blood into that tubular fluid or filtrate. This performs two important roles: elimination of hydrogen, or secretion of hydrogen, helps to control blood pH, and the secretion of potassium, ammonia, and certain drugs helps to eliminate these substances from the body. So reabsorption and secretion occur along the length of the nephron tubule and continues in the collecting ducts. This table compares the daily composition of substances in the plasma to their levels in the filtrate and in the urine, which is a reflection of the reabsorption of these substances. So assuming a glomerular filtration rate of 125 milliliters per minute, which works out to 180 liters per day, the total plasma volume is filtered several times, but 99% of that is reabsorbed, leaving to only 1 to 2 liters of urine being excreted per day. That fluid has to be replaced, of course, by ingestion of fluids, mainly also by water production at the cellular level. Proteins usually are not filtered, they stay in the plasma, but small amounts, up to 2 grams per day, may be filtered out because the filtration membrane is a little more permeable than your regular systemic capillary bed. So the filtered proteins are reabsorbed, mostly, leaving only 0.1 grams in the urine as a normal amount. Glucose is filtered and is 100% reabsorbed, again, based on normal glucose levels in the blood, so that 0 should appear in the urine. Urea is passively reabsorbed because it is small and fat soluble. So, although it is filtered, about half of it is reabsorbed passively, but it is also secreted so that approximately half shows up in the urine. Now, creatinine, which is a byproduct of muscle metabolism, is filtered but is not reabsorbed and is minimally secreted. So you'll see that the amount of creatinine that appears in the urine, 1.6 grams per day, is essentially equivalent to the amount that was filtered, and therefore creatinine clearance can be used to measure the rate of filtration, or the GFR. So we will now look at the different parts of the renal tubule and consider the processes of reabsorption and secretion in each area, also which substances are moved, and the general mechanisms for those movements between the blood and the filtrate. So the proximal convoluted tubule is the site of the largest amount of solute and water reabsorption. Most of the reabsorption occurs here in the PCT, and both solute and water reabsorption involves sodium. So on the basolateral side of the tubular cells, there are sodium-potassium pumps, or ATPases, that move sodium out of the tubular cells and into the interstitial fluid and blood in the peritubular capillaries. This creates a gradient where the concentration of sodium is low inside of the tubular cells. The tubular cells are cuboidal epithelial cells. They contain many mitochondria to provide energy for these active processes, along with extensive microvilli on the luminal side facing the filtrate to increase the surface area for reabsorption, as well as many embedded protein channels and transporters or carriers. These carriers include sodium symporters that use the gradient for sodium to also move glucose, amino acids, lactic acid, water-soluble vitamins, phosphate, and sulfate from the apical surface facing the filtrate into the cells. One of these transporters is the sodium-dependent glucose transporter, or SGLT. Once these substances move into the tubular cells, they then move along with sodium out of the basolateral side, usually by facilitated diffusion. So in the first half of the proximal convoluted tubule, sodium is co-transported with these substances and they are reabsorbed. Note that, as we said earlier, most escaped proteins and polypeptides are also reabsorbed here in the PCT, usually by pinocytosis, which is a type of endocytosis. Once these smaller proteins and polypeptides are inside the cell, they are broken down into their amino acids, which are then reabsorbed via facilitated diffusion out of the tubular cells into the peritubular blood. Here we have a diagram of some of the tubular cells from the proximal convoluted tubule, where their apical surface with the microvilli face the filtrate, or the fluid, in the tubule lumen. Shown in light blue is a sodium glucose symporter, such as the SGLT, which uses the gradient for sodium diffusion into the cell created by the pumping of sodium out on the basolateral surface. And this allows sodium to pull other molecules in this case glucose, making or allowing for glucose to be reabsorbed. As glucose passes through the cell, it is then moved out of the cell by facilitated diffusion on its basolateral side and is absorbed into the blood. This is how 100% of the glucose is reabsorbed, as well as amino acids and lactate or lactic acid. Almost all substances that are actively transported in this way have what is called a transport maximum. This is based on the number of carriers or transporters for that substance and speaks to the rate at which that substance can be maximally reabsorbed. If higher than normal amounts of substance are present in the filtrate, such as excess glucose that occurs with uncontrolled diabetes mellitus, then all the carriers will be saturated and the transport maximum will be exceeded, which means any excess substance will not be reabsorbed and will appear in the urine. Now, each substance also has a renal threshold, which is the blood concentration of that solute, above which the solute will begin to appear in the urine. The reabsorption threshold in the kidneys for glucose is around 200 milligrams per deciliter of blood. Now, sodium is not only reabsorbed by co-transport with the substances we noted before, but sodium is also reabsorbed while hydrogen is secreted from the tubular cells into the filtrate via sodium hydrogen ion antiporters. The secretion of hydrogen is tied to the reabsorption of bicarbonate via the carbonic anhydrase enzyme and reaction. We'll look at it on the next slide. Also note that bicarbonate that is present in the filtrate, or filtered bicarbonate, is reabsorbed in the PCT, most of it, and this is also coupled to sodium reabsorption and hydrogen secretion through carbonic anhydrase. So together, the secretion of hydrogen and the reabsorption of bicarbonate allow for regulation of blood pH. Now, once these substances are reabsorbed, it promotes the reabsorption of water by osmosis. So water will follow through the cells or between the cells and enter the peritubular capillaries proportionally. This, however, concentrates the remaining solutes in the filtrate and promotes their reabsorption by passive diffusion out of the filtrate through, or actually between the cells, and into the blood. This is how chloride, potassium, magnesium, calcium, and yes, some of that booger urea are reabsorbed as they follow their electrochemical gradients. Besides hydrogen, also note that urea and ammonia are secreted from the blood by the cells of the proximal convoluted tubules into the filtrate for elimination. Here we have the hydrogen and bicarbonate movements coupled to sodium reabsorption. So in panel A, or image A, the sodium hydrogen ion antiporter uses a gradient for sodium to move one molecule of sodium out of the filtrate into the tubular cell, while hydrogen is secreted and moved into the filtrate in the tubule lumen. The hydrogen comes from carbon dioxide, which either entered the cells from the filtrate or from the blood, or is produced by metabolic reactions. The combination of carbon dioxide and water, as catalyzed by carbonic anhydrase, forms carbonic acid, which dissociates into the hydrogen that is secreted and into a molecule of bicarbonate that moves out of the cell and into the blood by facilitated diffusion. Similar reaction occurs as depicted in image B for the reabsorption of filtered bicarbonate. So within the lumen, bicarbonate combines with secreted hydrogen, and carbonic anhydrase forms carbonic acid, which dissociates into water and carbon dioxide. So this is carbonic anhydrase located in the apical surface of these cells that faces the filtrate. Carbon dioxide diffuses into the cells and combines with water, forming carbonic acid and dissociated into hydrogen ion and bicarbonate, the reverse reactions. And this produces or reforms the bicarbonate, which is now passed out of the cells and into the blood by facilitated diffusion, and is again coupled to the reabsorption of sodium and the secretion of hydrogen. So, speaking of water reabsorption, there are two major conditions under which water can be reabsorbed in the renal tubules, either by obligatory reabsorption or facultative reabsorption. Both of these are forms of osmosis, where water is moving down its concentration gradient or following the high solute concentration. Obligatory reabsorption of water occurs when, for lack of a better word, water is obliged to follow the solutes because that region of the tubule is permeable to water, and water will passively follow the gradient created by the reabsorption of solutes. This is due to the presence of aquaporin 1 channels in the cells of the proximal convoluted tubule and descending loop of Henle. So these are the two parts of the tubule where obligatory, essentially non-modified water reabsorption occurs. However, facultative reabsorption describes variable water reabsorption, which is adapted to the body's specific needs at the times. And this is where water reabsorption is regulated by the levels of ADH targeting the kidneys and affecting the number of aquaporin 2 channels that are present in the principal cells of the distal convoluted tubule and collecting ducts. So facultative reabsorption of water is variable, where obligatory reabsorption not so much. This table attempts to summarize the reabsorption of specific substances in the proximal convoluted tubule. So again, sodium is reabsorbed by co-transport with several solutes as well as by exchange with hydrogen ion in the early part of the proximal convoluted tubule. And this results in 65% of the filtered sodium being reabsorbed here in the PCT. Water follows the solutes, so a proportional amount, 65% of the water is reabsorbed by osmosis. We saw that the co-transport with sodium was responsible for reabsorption of 100% of the glucose and 100% of the amino acids. Sodium co-transport was also responsible for reabsorption of large amounts of the bicarbonate. Note that both chloride and bicarbonate diffuse out of the filtrate into the tubular cells and are reabsorbed to balance the electrochemical gradient created by the movement of sodium out of the filtrate and into the blood. Chloride ion is passively reabsorbed along with potassium, urea, and the cations. Now, uric acid, which is a waste from nucleic acid metabolism, is actually 100% reabsorbed in the PCT, which seems like a waste for filtering it in the first place, but it's small and lipid soluble, so it's easily reabsorbed. However, it will be actively secreted later in the tubule. As the remaining filtrate passes from the proximal convoluted tubule into the loop of Henle, note that its composition is quite different from that of the original filtrate because now glucose, amino acids, and other nutrients are no longer present, along with large amounts of the electrolytes and water. However, the osmolarity, the solute concentration essentially, is the same as the plasma and initial filtrate because water reabsorption by osmosis was proportional to the reabsorption of solutes all along the proximal convoluted tubule. Now, the nephron tubule is unique in that it has selective permeability in the descending limb to water but is impermeable to water in the ascending limb, where in the ascending limb, the cells are permeable to sodium, chloride, and potassium, and this creates differences in not only what is reabsorbed, but also the concentration of the filtrate as it passes through the loop. Also note that within the medulla, the interstitial fluid surrounding the tubule becomes more concentrated deeper into the medulla. And these conditions allow the loop of Henle to form very concentrated urine when necessary. Also note that these conditions are supported by the flow of blood through the ascending and descending limbs of the vasa recta capillaries. So note that the descending limb is permeable only to water, and the water is pulled out by osmosis because of the high osmolarity of the surrounding interstitial fluid, so that by the time the filtrate reaches the hairpin turn in the loop of Henle, the osmolarity has changed from 300 milliosmoles to 1200 milliosmoles. The ascending limb is permeable to sodium and chloride and is actually pumped out in the thick portion. So to clarify, the symporter is a 1 sodium, 1 potassium, 2 chloride symporter that reabsorbs sodium, potassium, and chloride from the filtrate into the interstitial fluid and eventually the blood. However, the potassium tends to diffuse back into the filtrate so that the reabsorption essentially is that of sodium chloride. More sodium chloride is reabsorbed than water in the descending limb, so this greatly diminishes the osmolarity of the filtrate as it leaves the ascending limb and enters the distal convoluted tubule. Also note that the leaky reentrance of potassium into the filtrate creates an electrochemical gradient that promotes the reabsorption of other positive cations, including potassium and calcium and magnesium. Urea also sneaks in, but again is actively secreted in other parts of the nephron and collecting ducts. It's also important to note that the reabsorption of sodium chloride in the ascending limb helps to concentrate the interstitial fluid that promotes the reabsorption of water in the descending limb. And as the filtrate becomes more concentrated from the reabsorption of water in the descending limb, this also promotes the passive reabsorption of sodium chloride in the thin portion of the ascending limb. But again, in the thick portion, this is secondary active transport through the symporters. The net result is a dilute, greatly diminished filtrate volume entering the distal convoluted tubule. Another table attempting to summarize the reabsorption here in the loop of Henle. So sodium is reabsorbed by different mechanisms. We mentioned the co-transport with potassium and chloride, but also note that a significant amount of sodium diffuses into the blood via simple diffusion through the cells down its concentration gradient. There are also sodium hydrogen antiporters that allow sodium to be absorbed and hydrogen to be secreted in a thick ascending limb of the loop of Henle. This is most likely tied to the further reabsorption of bicarbonate that is also seen in the loop of Henle. Water is absorbed in the descending limb by osmosis, and then we have the symporters for sodium chloride and potassium. The recycling of the potassium created an electrochemical gradient that promoted the reabsorption of magnesium and calcium in variable amounts. When necessary, the mechanisms of urine concentration in the long loops of the juxtamedullary nephrons allow urine to be up to 4 times more concentrated than blood plasma, and this involves the effects of ADH, which contributes to the recycling of urea, which promotes water reabsorption as well as electrolyte reabsorption. But we also have the mechanisms associated with the anatomy of the loop of Henle and vasa recta that allow for concentration of the urine. All right, moving right along. By the time the remaining filtrate enters the distal convoluted tubules, about 80% of the filtered water has been reabsorbed along with most of the solutes. In this earlier initial part of the distal convoluted tubule, water, sodium, and chloride reabsorption continue. Water, of course, by osmosis, but sodium and chloride through sodium chloride symporters. So this accounts for absorption of another 10 to 15% of the filtered water and 5% each of the filtered sodium and chloride ion. Also note that the early DCT is a major site where parathyroid hormone stimulates reabsorption of calcium. So this is where calcium reabsorption is fine-tuned depending on the body's needs. Moving into the late distal convoluted tubule and collecting ducts. By this time, 90 to 95% of the filtered solutes and fluid have been returned to the blood. Note that there are 2 kind of cells that make up the tubule in this region. They are the principal cells and the intercalated cells. Under the influence of aldosterone, the principal cells reabsorb more sodium and secrete potassium. This secretion of potassium is the main source of potassium excretion in the urine, as in other parts of the tubule, potassium is mainly reabsorbed. The intercalated cells reabsorb potassium or bicarbonate in exchange for the secretion of hydrogen. Together, these help to regulate blood pH. So here in the late DCT and collecting ducts, the amount of water reabsorption and solute reabsorption, and in the case of potassium secretion, depends on the body's needs and is regulated by hormones.